Excited to share with you today the work that we've been doing on rhodopsin-associated autosomal dominant retinitis pigmentosa. Very briefly, Octant is an oral small molecule company building genetic medicines, drugs that will rescue the function of mutated proteins. Our lead program, OCT-980, is the first mechanism-based corrector therapy for RHO-ADRP. Unlike gene replacement or neuroprotective approaches, we're addressing the root cause of disease by directly correcting the toxic protein with the potential for restoring visual function. Roughly 20,000 patients in the United States and Europe are amenable, and with our genetically defined approach, we have a fast and efficient clinical development path. The key insight behind our program is really a shift from thinking about disease as a clinical label or even a gene label to thinking now about the targeting mechanism. Notably, rhodopsin is the largest genetic driver of autosomal dominant retinitis pigmentosa, but not all mutations are alike. We focus on a specific mechanistic class, misfolding and mistrafficking, where mutant rhodopsin misfolds in the ER, causing toxic gain of function. This leads to chronic ER stress and eventually rod cell death and retinal degeneration. It's a convergent, druggable biology where many mutations share a correctable folding defect. So how does it work? OCT-980 is an oral small molecule that reaches the retina and acts in the rod cell endoplasmic reticulum as a pharmacological chaperone. It stabilizes misfolded rhodopsin, restores folding across hundreds of variants, and relaxes the ER stress. Critically, it helps rhodopsin get out of the ER and into the outer segment where it can complex with 11-cis retinal. And importantly, OCT-980 is a 50-fold better corrector than it is an inhibitor, so that once rhodopsin reaches the outer segment, the drug is displaced by 11-cis retinal and therefore does not interfere with phototransduction. I just want to briefly show you why we're so excited about the program. What you're seeing here is data from a preclinical P23H knock-in mouse model, which faithfully recapitulates the human disease. What I'm showing you is that OCT-980 restores proper subcellular localization of rhodopsin. In diseased animals, you can see this in the second and third panel. Rhodopsin is trapped in the endoplasmic reticulum, and with treatment on the right, it can now reach the outer segment, matching wild-type animals. OCT-980 is correcting the trafficking defect. We also see extended survival of rod cells in the outer nuclear layer, and the correction of trafficking directly translates into structural rescue. And while I don't have time to show you today, we also have consistent improvement in functional features, such as ERG. This is, in fact, disease modification with translation from cellular correction to structural and functional rescue, underpinning our confidence in going into the clinic. Using our synthetic biology approach, we've comprehensively tested every single amino acid substitution across all positions of rhodopsin. And these heat maps show every variant, untreated on top and OCT-980 treatment on the bottom. Red indicates the protein trapped in the ER, and white trafficking at the levels of wild type. And I hope you can appreciate that 980 is a true pan-variant corrector. It addresses the large majority of misfolding and mistrafficking variants, and the residual red that you see is the orthosteric binding site and the few positions like disulfide bonds which massively destabilize the protein. This has allowed us to define a treatable molecular subset of disease rather than chasing individual variants. Overall, we estimate correction of approximately 60% of the total rhodopsin-associated disease burden. We're currently in the process of finalizing our phase 1A healthy volunteer study, and correct RP patient dosing will begin in September. In addition to safety, tolerability, and PK, our disease endpoints are directly linked to rod cell biology. We'll measure EZ width, EZ area, re-expansion as structural markers, and low light visual acuity improvement for functional assessment. Outer segment renewal occurs every 10 to 14 days, and as such, we believe that we're going to see meaningful changes within just 12 weeks of therapy. Finally, I just want to thank the exceptional group of advisors and investigators, as well as the FFB and Retina UK that have supported the development of our clinical development plan. We believe 980 has the potential to redefine how we treat inherited retinal disease by correcting the underlying biological mechanisms of disease. Thank you.